with Dr. Marie Christine de Vernagel. And after that, he came to Texas and worked with someone that I admire greatly and had the pleasure of knowing, Dr. Greg Mundy, who is really a pioneer in bone biology. And Gabriel did some fantastic work there with Greg and uncovering, and I think this is maybe where I first discovered your research, thinking about members of the TGF-beta and BMP signaling family and their role in the skeleton, as we were colleagues kind of coming up together at that time. And then after working with Greg, he went into biotech and had a very successful career looking for new therapeutic targets of inflammatory arthritis at a number of high-profile companies that you'll recognize: Osiris, Pharmacia, and Pfizer. But then decided to come back into academia, and he brought that perspective of working at the translational and clinical interface back with him to academia at WashU. And he joined the Division of Bone and Mineral Diseases there, and in 2010, and is now a professor in medicine. So Dr. Mbalaviele is a leader in studying the inflammasome and its role in bone biology and in skeletal disease, with particular expertise in innate immunity. He has over 70 papers, many of which are highly comprehensive studies that are in high impact journals like eLife and PLOS Biology, where he's uncovered these cellular and molecular mechanisms by which the inflammasome is causing things like osteomyelitis or exacerbating infection and bone resorption in these pathological scenarios. And I think today we'll get to hear a little bit about his work on pyroptosis and gasdermins in these processes. So in addition to his scientific accomplishments, Gabriel is also serving in a number of capacities, including at the ASBMR and as a service on standing study section member at the SBDD study section, as well as on the VA special emphasis panel on endocrinology. And in addition, he has leadership roles at WashU, where he co-directs the Forum of Underrepresented in Medicine, and is also bringing that industry expertise to bear in helping other faculty to reach out to the biotech community through Bio St. Louis Biogenerator. So Dr. Mbalaviele, it is truly a pleasure to have you here with us, and we're looking forward to hearing your seminar on parsing the paradoxical, pyroptotic, and osteoclastogenic fates of macrophages.
Okay, so let me share my slide, and
looks great.
Okay, and wow, thank you so much, Tamara, for that introduction. Okay, really, I think you should give this a talk. I was so impressed. Thank you. I really feel good, and really thank you very much for inviting me. And I really enjoy the visit, even if it's a virtual. And I have a meeting with a PI this morning with a trainee, and a lot of great science going on. And thank you very much. And I would like to use this opportunity, to thank Mary Nakamura, really for her support behind the scene. I really thank you all for joining this meeting. Okay, and okay, let's get started. So this is my disclosure. Pyroptosis is a form of inflammatory cell death driven by a family of a protein called gasdermin, in short GSDM. And shown here are are member of this family in a human and in a mouse. Can you hear me?
Perfect.
Great. So pyroptosis is induced by a couple of pathways, including the inflammasome. And this form of cell death takes place in various cell types, including the one shown here, epithelial cells, neutrophil, and macrophage. So macrophage, undergo pyroptosis in inflammatory condition. And we also know that macrophage differentiate into osteoclasts, in inflammatory conditions such as rheumatoid arthritis. Okay, and for some of you who may not be familiar with bone biology, so the osteoclasts are multinucleated cells, and they are responsible for bone resorption in a normal and disease condition. So we have been working on this pathway for some time, and the question that we are asking now is how can a macrophage undergo pyroptosis and differentiate into osteoclasts under the same inflammatory condition in which they die? So for you to, to help you understand, appreciate the sense or, the rationale of this question, to understand to understand a little bit where we are going with our research. So I have organized, my presentation in three parts. So in part one, I will give a brief background on inflammasome and pyroptosis. In part two, I will briefly review, the literature on the role of inflammasome and pyroptosis in skeletal metabolism and disease. And in part three, I will address, try to address this question around macrophage, pyroptosis, and osteoclast differentiation in inflammatory condition. So starting with the first part. So the inflammasome are intracellular protein complex. This complex comprises a receptor which recognize microbial product known as a pathogen-associated molecular product, in short, PAMPs. They also recognize non-microbial product known as danger-associated molecular product, DAMPs. And this receptor can also be activated by gain-of-function mutation, and I will come back to this in the next slide. So there are several inflammasome receptors. Shown here are the most studied one. Okay, they include NRP1, NRP3, and so on. So I apologize for the abbreviation. Okay, so the complex comprises, a receptor of inflammasome, and a receptor, and an adaptive protein called AC, which bridge the receptor to caspase 1. And caspase 1 is the catalytic subunit of the complex. It's the enzyme that processes the substrate of the inflammasome. Actually, the inflammasome, just very quickly, they are named after the receptor. So this is what they are referred to as the NRP1 inflammasome, NRP3 inflammasome, and so on. So shown here are what are known as classical or canonical inflammasome, and non-canonical inflammasome have also been identified. So for today's presentation, because of time limitation, so the discussion presentation will revolve around the NRP3 inflammasome. So what is the function of the inflammasome? Like I said, so they recognize microbial and non-microbial product. So upon recognition of a microbial product or or microbes, they are involved in innate immunity. Okay, they are first line of defense against pathogen, and they are also involved in tissue repair, like I said, in context of infection or even non-infectious condition. So I think really the function of the inflammasome are to restore tissue homeostasis after injury. But it's when they are overactivated, they are not controlled, that they can lead to to inflammation of a high grade, and this can lead to disease. So the question is, how do the inflammasome accomplish? I'm sorry, I have to move some of these things here. Okay, so how do the inflammasome accomplish the function? Oh, the slide is stuck. Cannot move. Just cannot move the slide. Just 1 second. Try to understand what is going on. It says the screen sharing is paused.
Oh, seems throwing us new challenges.
Yeah, this is what happened with. It said it's a pause. Screen sharing is a pause. Should I stop and maybe start it again?
Maybe that's a good idea. Mary and Pui, if you have any insights, then.
Okay, so let me start to. Yes, I know this part of. Okay, this share again, and Okay, here we go. Okay, so the question was, how do, of course, I'm not going to touch on some of these functions. So how do the inflammasome, mediate the function? So they do so by mediating, regulating the maturation of a cytokine, of the cytokine shown here, interleukin 1 beta, in short, I1 beta at 18, and by, causing pyroptosis. Okay, so unlike some other cytokines that we know, like over inflammatory cytokine like TNF alpha, IL-6, so actually this cytokine, they are produced as inactive cytokine. They are produced as a pro-cytokine. Okay, and they don't have a signal peptide for secretion through the ER-Golgi pathway. So when they are expressed, produced, they stay in the cytoplasm. So their activation and secretion require the inflammasome. So what happens is when the inflammasome receptor, like I say, can be NLP3, can it recognize the activator, the ligand. So it oligomerizes, then it recruits this adaptive protein that I mentioned, AC, and if this interaction occurs through some identical domain, it's a homophilic protein-protein interaction. You can see this domain, which is called parin here, is very similar to the domain of AC. So through this, oligomerized receptor, recruit AC, which oligomerized now recruited procaspase 1. So forming this complex here that you see this prion-like structure, which is the bacterial state of the inflammasome. So in this process, for some unknown and clear mechanism, so procaspase 1 is activated, and here is released. Then activated caspase, active caspase 1 process this procytokine into the active form. Okay, but it was not known how these cytokines are secreted. It's only about 7-8 years ago that it was discovered that caspase 1 in parallel, also in a parallel of processing, this procytokine, it also cleave this protein, okay, gasolamine D, member of the family that I mentioned, in short, GSDMD. So gasolamine D has two amino acids of a family member. It has two domains, and it is in autoimmune conformation. So when it's cleaved by caspase 1, the N-terminal domain translocated to the plasma membrane, where it forms pore through which, there's a pore through which the cytokine are secreted to the extracellular environment. So cell like neutrophil, macrophage, and epithelial cells that I mentioned, they can produce this, can do this job, produce this cytokine while they are still alive. So in the field, they are referred to as hyperactive cells. So cells can do this and being alive. What happens is, if there is overstimulation of the pathway, chronic activation of hyperactivation or chronic activation of the pathway, then this can lead to excessive pore formation by gasolamine, as you can see here, as depicted here. And this can lead to, water getting in through this pore, causing osmotic shock. And this can lead to cell lysis, cell rupture. So this form of lytic cell death is called pyroptosis. Okay, so pyroptosis is very inflammatory. It's highly inflammatory because in addition to the cytokines, R1 beta and 18 that are to be released to the extracellular environment, other intracellular content are also released, and some of them can be pro-inflammatory, like the one shown here. Okay, so this is the first part of the presentation. So what I have, to summarize what I have said so far. So the inflammasome, they are a family of intracellular protein complex. They are activated by microbial product and non-microbial product, which can be endogenous or exogenous. And they can also be activated by mutation, and I will show this in the next slide. So they are involved in the maturation of this cytokine, and they can cause pyroptosis if there is overstimulation or chronic stimulation. They are involved in innate immunity, in tissue repair, but it's excessive response or uncontrolled response that can lead to, to high inflammation, high grade of inflammation and disease. So moving to the second part of the presentation on the function of inflammasome and pyroptosis in skeletal homeostasis and disease. So this is a very busy slide, okay, and I apologize for this. So this slide, it's a show, the, you know, various skeletal disease in which component of the inflammasome, like in parentheses, like NRP3, M2, NRC4. So the component in which disease in which the component of the inflammasome have been shown to be involved. Okay, and because of the time, so I'm not going to go through this. And actually, we have, we published a review a couple of years ago, and about 3 years ago, in which we review the role of this pathway in skeletal disease. So what I'm going to do is just to elaborate a little bit, explain a little bit on skeletal complication associated with mutations that cause constitutive activation of of the um, the receptor. So something that I didn't say in the previous slide is, for example, this mutation in the receptor of the inflammasome like NRP3, they are believed to cause constitutive, change of the conformation of the protein leading to constitutive recruitment of, the component of the inflammation that I described. So it was discovered about 20 years ago that in human, that NRP3 activating mutation causes a spectrum of autoinflammatory disease known as cryopyrin-associated periodic syndrome, in short, CAPS. So CAPS are a rare autoinflammatory disease, they're caused by autosomal dominant mutation, in this case, in NLRP3. And then the phenotype range from severe, from mild, mild, patient with mild, moderate, to severe phenotype. And the severe phenotype is called NOMID, which is a neonatal onset multisystem inflammatory disease. So, and so in general, like I say, it's a spectrum of disorder. They are associated with, caused by, driven by the excessive production of I1 beta 18. Patient develop a skin rash, like shown here, and they have episode of a periodic, periodic fever event. So, and NOMID, okay, the most severe phenotype, is associated with skeletal abnormality, as shown, skeletal abnormality, CNS issue, even early death. So shown here, this is a normal knee of a normal patient, and the knee of NOMID patient, okay, at age 3 and age 9. And I hope that you can see, you can appreciate that at this age, there are some lesion in the epiphysis of the distal femur. You can see here, start at this age, then the lesion get worse with the time. Okay. So, like I said, CAPS are a rare disorder, and because of this, so the access, as you can imagine, the access to sample for study is limited. So this really provides an opportunity, a rationale for, genetic mouse disease model, of this disorder to understand advanced knowledge on the pathogenesis of this disorder, and also with the expectation that the knowledge gained from studying this, this rare disease can be applied, to inflammatory disease that affect the general population. So Al Hoffman from UCSD is a physician scientist who will see CAPS patient. Just to make sure, everything, can you hear me well?
Yep.
Great. Yes. Okay. So Hal is from UCSD. Is CAPS patient. So and he's a scientist. Physician scientist. Sorry. So he generated a mouse model to mimic a human disorder. Okay. And in this case, the allele, mutated allele of NRP3 were flux, so you can use a crew of interest to activate the mutation. And so here are the, like I said, it's a spectrum, mild, moderate to severe, and these are the mutations that are found in a patient. And this mutation here is in a NOMID patient. So he generated this mouse, and he was generous enough. So this is when I joined, like Tamara was saying, when I came back to academia. So I was looking for something to do. I didn't have a project, and I was reading papers, so I came across this paper. And so I touched base with him. He was generous enough. He has this mice, but he has not characterized this one. And he just shared this mice with us. So this is what really what launched my career. And so we studied this mouse model. We characterize it. And well, we did a lot of study, and shown here is just one of the, the approach that we use, where again, I say the mutation is a flux, and you breed with your Cre of interest. Here we use lysozyme M Cre for activation in myeloid cell, like monocyte macrophage and dendritic cell. And then we got wild type and no-meat mice. What you see here are wild type and no-meat mice. And you can appreciate that no-meat mice, develop skin inflammation. And I'm showing this because in human they have rash, but in the mouse at least you can see skin inflammation. So these are 3 weeks old mice. And so, I mean, let me slice to summarize, the work of, for 7 years or several years of work. And so what I show here is, in this mice, just like a human, they have, inflammation. They develop inflammation, high level of R1 beta. This is serum level of cytokine. Same thing for R18. You compare wild type to mice. They have this. So they have inflammation, systemic inflammation. They have, leukocytosis, high, count of white blood cell driven by neutrophil. So they have a neutrophilia. So really, to summarize this work, so and again, it's R1 beta driven inflammation. Mice have short lifespan. Also, they died on the 3 weeks of age. They have CNN issues, skeletal abnormality, low bone mass. In a patient, these are not well studied, so we show that as you would expect with all this inflammation going on. So low bone mass driven by excessive, osteoclast formation. And like in a human, again, I didn't have time to elaborate what is happening in a patient. Patient have disorganized growth plate, and we see things in a mice. And but something that we did not really observe, which was very disappointed, is we didn't see bulky epiphysis like what happened in a patient. And we have some speculation, and we can talk about this if you are interested. So we published this, this work this study in a couple of paper, and some of the data were produced by other investigators, such as in this paper, in which they use another no-meat model and also found bone abnormality. Okay, so and okay, so far good. So it looks like in no-meat in this mice, you have a high level of R1 beta, R18. You have disease. So then the question is the action of the inflammasome limited to just production of the cytokine? Okay, and the answer is no. And as you can see here, actually it was, reported, in a clinic that a skeletal lesion that I described with Nancy, actually they continue to grow in a patient, in no-meat patient, even when they are on R1 blocker, like for example anakinra, which is a drug, it's a R1 receptor antagonist. Okay, so an antibody, I think they show that the lesion grow, not to regress, but to grow when patients are taking this drug. And Al Hoffman, use a mouse anakinra, and they also showed in the mouse model that when you block use of the drug, you only partially inhibit, the symptom of a capsule in the mice. And he went on again to do even study where delete, the gene, the receptor, R1 receptor, this is cytokine, and show again in the mouse model that they see residual inflammation in the mouse model. Okay. Then the hypothesis based on this finding was that pyroptosis may be responsible for this outcome. So this makes sense because, like I told you, pyroptosis, if a cell undergo pyroptosis, so you have release not just of R1 beta 18, but also over, inflammatory factor, like some shown here, like S100A protein, eicosanoid and prostaglandin. Okay, so one way to test this hypothesis, to prevent R1 beta 18 release and pyroptosis is to delete gastramine D in this case, okay, because gastramine D is downstream of the inflammasome. And this is what we did, okay. And shown here, wild type mice, no-meat mice, we again, yes, no-meat mice and no-meat mice lacking gastramine D. So again, to remind you, in no-meat, so you have a constitutive activation of NLRP3. So it forms the mutation, make it a protein to recruit over component of the inflammasome independently of the ligand. You don't need the DAMPs, you don't need the PAMPs. Okay, but if you have them, you have more response. So, and so what we found was that, so you can see these mice have skin disease, inflammation. And these mice here lacking glycemia D, they are just normal. Just like a wild type, we monitor them for more than a year, they normal. No bone phenotype, no organ damage, no inflammation. Okay, we published this in 2018. In the same year, another group now working on a different model of inflammasome-driven disease. This time it's the model of familial Mediterranean fever. Okay, in this case, the inflammasome, like I said, there are several receptors, and the receptor here is parin. And again, like NLRP3, there are mutations which cause constitutive activation for parin. Okay, they also did the same thing. I mean, we didn't know what each lab was doing, just published the same year. And we also showed that mice, again, these mice, they are sick, have a skin lesion. And then when they lack glycemia D, they are just normal. So I think really the impact of this work was really has some impact in the field. Actually, it triggers some interest in trying to find a drug to block glycemia D, maybe to treat, inflammasome-driven disease. So looks good, very good. So it looks like, inhibiting glycemia D would be a mechanism to treat inflammasome-driven disease. But one of the person in my lab, Dr. Chen Wang, she just wanted to know what will happen. These mice, they look normal, but what will happen if they are challenged with something? If they just like wild type, what happens if they are challenged? So, and I'm sorry. Yes. So this is what she did, and she took mice at around this age. But I think for the no meat, they were used a little younger because they died earlier. So I treat them with LPS or with PBS or LPS, and what you see here again, dark black is a white type, blue is a glycemia D knockout, red is no meat, and purple is a no meat lacking glycemia D. So this is what we found. So high level of cytokine in the no-meat, R1 beta, same thing with R18. So this was expected to what we published before, actually. And then this is a block. You don't have this secretion when you don't have glycemia D. So very good. And then we treat with LPS, now wild type, okay. Wild type, you treat with LPS, start to reduce, production of the cytokine. And in a no-meat, okay, here start high, but if you add LPS, you get even more cytokine production. Again, this was expected, okay? And now this is what we did not expect, okay? Hope that I'm not confusing you. This is what we did not expect. So that now in this no-meat mice, okay, which are really normal, but if you treat it with LPS, actually there was no change, no decrease in our 1 beta level, with a little bit of decrease in our 18. So we were really expecting that this mice, the level will be here. Okay, so we say, okay, this is LPS or it's just specific to LPS, this bacterial product. So, and we use TNF alpha, we found about the same trend. So, the conclusion of this work is that, okay, these mice, the no-meat mice, the inflammation with inflammatory stimuli can overcome the protective effect of glycemia deficiency that we see in no-meat mice. So, ready to summarize this part of the presentation. So, what we found is in mice. So, like mice, no-meat mice, like a patient, they have excessive production of R1 beta, R18. They have low bone mass because of increased osteoclast differentiation. This is undergo pyroptosis, and we think that is driven by glycemia D, okay, in no-meat mice, because what happened is if you delete glycemia D from this mice, now everything is normalized. You have almost no to very negligible inflammation. Okay, but we think that these mice have an underlying condition, like a human, okay? The underlying condition is a mutation in NLRP3. So even if they are normal, in a normal condition, but if they are challenged, now they develop a disease, they develop systemic inflammation. Again, I didn't show the data. Yes, I showed the data that I show, and they have pyroptosis. And here in this case, because they lack glycemia D, we found that it's a glycemia E, the other member of the family, that is involved to some extent, not completely. It does not rescue. If you delete glycemia E in this mice here, here you delete glycemia E, mice do not go back to normal. So there is some residual inflammation. Okay, we didn't look at bone or osteoclast, but there is no reason to believe that the osteoclast will not be affected here. Okay, so I think we are good. That is the second part. Now moving to the third part, which is the last part of the presentation, which is really our our question around the macrophage pyroptosis and osteoclast differentiation in inflammatory condition. And here are this I'm going to go over our unpublished data. What I have shown is most of the things that I just described were published, and here are unpublished data, very preliminary. Okay, so and before, so let me refresh your memory. Actually, tell you something that I didn't really say very clearly. So actually, the activation of the inflammasome pathway, it's a two-step process. Okay, two-step process, especially for this inflammasome for NRP3 inflammasome. So there is what is called priming, step or the priming signal. Okay, and these are signals that are induced by pathway like LPS pathway or cytokine TNF, even R1 beta as a feedback mechanism, or positive feedback mechanism. So, and this signal, okay, we go through pathway like NF kappa B. Actually, we probably a couple of years ago that P30 MAP kinase is also involved. So what this pathway do is to induce, the induced expression of this pro-cytokine, pro-R1 beta, induce the expression of NRP3 and the expression of galectin D, because the level of this factor are kept low to prevent, spontaneous unwanted inflammation. But if this one are not really inducible, positively expressed. So priming signal is really to induce the expression. This is why I say NRP3, because its expression is inducible. And so you have this, but nothing happened. Then you need a second signal. So this is where I was talking about DAMS, pumps, or mutation. This second step is now for the assembly of this complex. Okay, what you see here, NRP3, oligomerizole. If you look at this very closely, you will see how you will figure out. So they do and they form this. So and this assembly signal are induced by a signal like for example, in the case of NRP3, by this inophore, this is a bacterial product. So it causes perturb the membrane, the plasma membrane, and this is sensed by cells and activate, the inflammasome because you have ion fluxes, effluxes. So, like I say, it could be also by mutation, like I showed in the case of NRP3 and Parsing. Okay, so you need these two signals. Then this, like I said, lead to the activation of a caspase 1, then, processing of pro-cytokine and cleaving galectin D, then downstream signaling. But for the work that I'm going to describe, so instead of using, say expressing mutated NLRP3, but we chose to work with wild type cells so we can stimulate them with LPS and then with something like a nagericine, okay, with the ionophore, to activate the complex instead of having the complex being active all the time, okay. But we can again talk more about why, the reason for this. So this is the system that we use going forward in the study that I'm going to describe. And so if you take macrophage, okay, wild type macrophage. So this is what we did here. Isolate wild type macrophage from bone marrow, and I treat them with, if you treat macrophage with LPS and looking at the one beta release, there's almost nothing. Okay. Human situation is a little bit different, but in the mouse, there's nothing. You treat with nagericine, nothing, but not shown here. Now, if you combine, these two stimuli, LPS and nagericine, you have release of R1 beta, excessive amount of, and the release of LDH. So LDH is a lactate dehydrogenase. So this is a marker of a pyroptosis in vitro, because and the pore. Okay, something that I didn't really, I forgot to mention this. So the pore that I show you of a galectin D, so this pore only let go through small molecule like R1 beta 18 because of the molecular weight is very small. Okay, so there's really no selectivity for thing to go through the pore. It's just the size. So R1 beta 18 around 18 kilodalton they can go through. But this one, LDH, the molecular weight is more than 45 kilodalton. So the only way that it can be released, at least is if a cell membrane, there is a perturbation, there is a cell membrane rupture. Okay, so if you use the ligand, the activator of the inflammasome, then you will use this as a readout of pyroptosis. But if you use a apoptotic, similar will be different story. Okay, so this is what we see. Okay, when you activate with LPS and adjacent, and so what this show is, if you take a macrophage, you can induce them to become pyroptotic. Okay, like I said, and we know that the macrophage can be induced to differentiate into osteoclast, and I will show this in the next slide. Okay, so then our famous question, going back to this, so the question is, how can this happen? So macrophage undergo pyroptosis and also can differentiate into osteoclast. So how this happen? How can this happen? This be possible? And one hypothesis, so we have several hypotheses, again, for the sake of time, just I'm going to discuss this one, which is that actually the inflammasome pathway is just simply down-regulated during osteoclast differentiation. So, and this is why cells do not go this way, and this goes this, because they don't undergo pyroptosis, and so they can differentiate. So actually, I mean, like I said, we have several hypotheses, and this one, actually, there are evidence, a free line of evidence to support this hypothesis that this is really the case. Okay, and first evidence is shown here. Okay, like I said, you take a macrophage, and you just add this cytokine or rancor. It's a very potent for driving also macrophage into osteoclast. Okay, so multinucleated cell. Okay, so then we look at it. So what happened to the inflammasome pathway? We look at the very component of the pathway, and I'm just showing NLRP3. So what do you see? So we treat the macrophage will be here day 0 with rancor for several days, and so usually the osteoclasts are formed by day 3, day 4, and so you can see that during osteoclast differentiation, the level of NLRP3 decline, go down, and if you let the cell go further, then the level of NLRP3 will decline further, will be almost undetectable. So expression of NLRP3 and the expression of NLRP3 go down during osteoclast differentiation. Now, the next experiment that we did now is to take macrophage osteoclast similar to with LPS and treat with this, with nigericin. So here to induce whatever can be induced, and here with nigericin to form the complex. And here we are looking at now the activity of the complex, looking at the cleavage of this and the secretion of R1 beta. And and this is what we see here. Macrophage will be something here, day 0, and osteoclast sometime, around day 4. Okay. So what we found is during osteoclast differentiation, actually the level of glycemia D go up. This in contrast to what we see with NLRP3. And right now we try to figure out what is the meaning of this. In any case, so you have macrophage, you have osteoclast, and you treat it with LPS or nigericin, nothing happened. You're looking at the cleavage of glycemia D. And if you combine both stimuli, you have glycemia D cleavage, generating this N-terminal fragment. And actually, it's this N-terminal fragment that to form pore. Okay, you see this in here. We see also the same thing in the macrophage, but as you can see, but the extent of cleavage is lesser in osteoclast compared to macrophage, despite high level of glycemia D. And so just looking at glycemia D, this is the cleavage here, high about the secretion of R1 beta, and what is shown here, you have macrophage with both stimuli, nice production of R1 beta, but this production is decreased in osteoclast. So, and if you measure, measure pyroptosis, and you see the same thing, decrease high in macrophage, down in low in osteoclast. So the conclusion from this work is that the inflammasome pathway, NLRP3 inflammasome pathway, is down regulated during osteoclast and differentiation. I mean, supporting the hypothesis. So this evidence 1, and 2. Here we looking at really the assembly of a complex. The complex itself, okay? This complex here, as you use LPS and nigericin, then you form this complex. And here the readout is just this complex. So here we took advantage, really to make the story short, of this mouse reporter called AC. So AC is the protein that I mentioned to you, but the people engineer AC Saturn, conjugate this with fluorescence fluorescent peptide. Okay, instead of using antibody, you can use this reporter. So when you activate the inflammasome, if the inflammasome are formed active, then you can see this as aggregate, as a punctate. Okay, and they are referred to as AC spec in the field. And so this is what we did here. We take osteoclast culture, we treat it with LPS. Okay, and I cannot see the bottom. Okay, anyway, and so these are osteoclast culture, and stain in blue with a dot P. Green is this AC citrine, so all cells supposed to be green. Okay, I mean because they are macrophage and they derived from macrophage. These are macrophage culture. Sorry, that I'm missing some detail. So treated with macrophage treated with rancor for about 4 days. Okay, and so when you treat in also classic culture, they are the culture are always heterogeneous. So you have said that are undifferentiated macrophage. It depends on the time that you stop the culture. So here you have macrophage which are have not differentiated. I'm sorry. Let me back off a little. So to say yes. So say we're treating with rancor, macrophage rancor for 4 days. Then let's treat it with LPS. Then so green is a citrine, blue is a DAPI, and red here we stand with the integrin alpha, alpha 3 beta 3 integrin, beta 3 integrin. So this integrin is upregulated during osteoclast differentiation. Okay, during osteoclast differentiation, and so what I was saying is that this culture is heterogeneous, culture, and so you have undifferentiated macrophage, which they are just green here. Then you have, cells that are differentiated into the osteoclast. They are still mononucleated, but they start to express the integrin, beta-3 integrin here, and there's another cell with 2 nuclei, so because they fuse with time, and they also express the integrin, and this is another cell with 3 nuclei. And actually, when you do osteoclast stuff or work, actually this one is considered as osteoclast in vitro. I mean, it's an arbitrary cutoff. Cell with 3 nuclei is considered as osteoclast. So, and what you see is this. Okay, nothing. I mean, you see the AC citrine. The AC is just diffuse. Now, treat with nigericin. It's a compound which causes the activation of a complex. Now, what you see is now this AC that it was diffused, it become from aggregate or spec, and you see that almost all macrophage are have the spec. Okay, we see this which was expected, thinking that we discovered or known in the literature. But what we didn't see is in cells that are beta-3 integrin positive. You see them here, and there is no spec, not because of the red color. Okay, not the precursor, not the osteoclast with this with 3 nuclei here. And this change, the morphology could be maybe because of this ionophore, so we don't know. But in any case, so no spec formation. Again, the conclusion is that of the osteoclast lineage, okay, the osteoclast lineage does not assemble functional NLP3 inflammasome. Okay, and then the third evidence is now looking at again this pathway similar with LPS, nigericin, and just looking at this time the readout is the entry of PI, propionidine iodide, through this pore. And very quickly, running out of time, and again in blue is staining. Red is a PI, and then purple is a merge image. So, and what you see here, then you have the osteoclast for you. The osteoclast will be here, one osteoclast, one cell with main nuclei, the osteoclast. So what you can see is, PR positive are mononucleated cells, but if you look at the osteoclast, they are negative. Sometimes you see a signal, but it could be cell on top of the osteoclast. And this is really glycemic D dependent because in wild type, you can see this, but in cell lacking glycemic D, there is no PR uptake. Okay, so all of this data taken together, led to the conclusion that NLP inflammasome pathway is down-regulated during osteoclast differentiation, and we see this at and for example NLP3 expression level is decreased, and even the efficiency to form of the complex is decreased in during this process. Then the question is, okay, what down-regulate the pathway, at the translational level? What down-regulate the pathway at the complex formation level? So we use transcriptomic and proteomic approach to do this, and so to try to address this question, just to show you a piece of data very quickly. Like I said, all these are published. It's an ongoing study. So here we use RNA-seq, looking at a macrophage, treated with LPS, a sample RNA triplicate, and PBS, LPS, then macrophage, osteoclast, again osteoclast with macrophage treated with rancor for about 4 days. So we identify, a transcript called SIMPO2, which, as you can see, was only expressed or upregulated in osteoclast treated with LPS. And we confirm using qPCR that only osteoclast with LPS expresses this protein, that means this gene. So, and so what is a SIMPO2? It's something that is involved in actin polymerization. Nothing known about the osteoclast. So, and we are looking for something that is expressed maybe down-regulated to the inflammasome. Is this what SIMPO2 does? We don't know. So, and we are, like I said, this is just to show you what is going on in the lab. And then the final part, just in a few minutes, is the question about, now we just try to look what affect maybe the complex formation by the inflammasome. So I'm going to skip the detail because of the time, but if I can explain. But the bottom line is we use this approach, we use this strategy. We did IP using antibody that only recognize BS1, AC, conjugated with cytosine. Actually, so here has a HA tag. So we want to immunoprecipitate what come down with AC. So this one will be used as a negative control. So we identify this protein. We use a mass spec and identify this protein called LAMTOR4. Okay, then we did confirm doing IP. We think that LAMTOR4 associated with AC. And then ready to make sure that this is really true. And we did, instead of IP, we also did a proximity ligation assay, which very briefly, you have two proteins, red and green. You have primary antibody that recognize this protein. Then you come up with secondary antibody conjugated with oligonucleotide, which are complementary. Then you can do PCR reaction with fluorescent primer. And if it works, you can see a signal. And so we use here LAMTOR4 with protein that we identify with AC. And we see the signal, which is a really red signal that you see here. So green is AC. But then if both proteins are together close enough, you can get a signal that you see in red here in osteoclast, but even also in osteoclast precursor. But you don't see this if you miss one of the antibodies, for example, if you use isotype IgG. And LAMTOR4, what is it? So it's a protein that is really involved. It's a component of the regulatory Riger complex involved in cell metabolism. And we just wonder, so is this protein, what happened to it during osteoclast differentiation? Bottom line is no change in the protein, the expression of this protein in this different conditions compared macrophage to osteoclast. So while we were doing this work, and then two papers came out just last year. We started doing this work about 3 years ago. And we are not very as fast as that. So the two papers published showing that this protein is involved in vitamin D maturation, but different level, not where we see interaction with the pathway. But anyway, so we're looking for something that inhibits the pathway, but why is this has to do with inflammasome inhibition? So we don't know. So all these studies are underway. And so to recap this part, and so what I told you is that the inflammasome pathway is down regulated during osteoclast differentiation at both a priming and a complex formation level. We really don't know what are the mechanisms, and hopefully we will figure out. And so why do we care? So why do we care about inflammasome pathway going down during osteoclast differentiation? And we think it's because if we can identify a factor, it's a long shot, that don't really inflammasome, in the osteoclast lineage. So maybe that factor can be used to prevent inflammation in the disease. And like Tamara mentions, actually now we have NH funding. We have we fund from NIH, this in collaboration with device where we are looking at the pathogenesis, protein descent pathogenesis of osteomyelitis. And our hypothesis is that the inflammasome are involved in that disease because, for example, NLRP3 is downregulated. So we think that this study can help us really try to understand some mechanisms of OM pathogenesis. So now, since you use this is the last slide, and the question about paroptosis and also class differentiation, actually we think, really based on what I just showed, that the paroptosis and also pyroptotic and osteoclastogenic effect of macrophages are not paradoxical. So what we think is going on, again, this working model is that your inflammation, so you have cells that express high level of inflammasome, like macrophage over cell like neutrophil, but some of the cells, it's a high level of inflammasome, they undergo paroptosis, like shown here, they release cytokine like I1 beta and other, inflammatory mediator over DAMPs, which act on, lymphocyte, mesenchymal cell to release, I cannot see the bottom of my slide anyway. So then there's a factor, act on this cell to produce some other factor like a rancor, some local factor like a rancor. Then this, and this factor actually drive the expression, downregulate the inflammasome in some cells that did not undergo paroptosis, okay, like this, decrease the inflammasome, level of the inflammasome activity and paroptosis, then ultimately this leads to osteoclast formation. So, and with that, I'm a little late, and just to acknowledge people who did the work in the lab, and the action here, and here are my collaborator at WashU, and collaborator outside of WashU, including Al Hoffman from UCSD. Okay, with that, so I'm ready to take your question. Thank you.
Great. Thank you so much. Questions? Dan.
Yes.
That was really a fascinating talk, talking about a pathway I don't know much about. But it turns out that in clinic, I was presented with a 28-year-old who developed this multisystemic inflammatory syndrome from COVID. What you were talking about at the molecular level is, I'm certain, what was going on. He had his heart was involved, his severe myocarditis, his pancreas was involved. That was what was interesting, endocrinology, and with sky high diabetes, wildly out of control, levels that I've never heard of before. And his joints were involved. So it really was multisystemic inflammatory syndrome triggered by the COVID-19, which is clearly the PAMP, if you like, which got the whole process going. And I'm wondering if you've had occasion to study any of these patients who are triggered by an external stimulus like that.
Yes, absolutely. Very, very good point. Yes, no, I didn't have a chance, but I know that that is already a cytokine storm that occur in some of the patients, like you say, in COVID. I mean, some of the patients, I don't know, 10 or 20% of the patients, they develop this cytokine storm. Absolutely. So the inflammasome have been shown to be involved, associated, especially with NLRP3, even another inflammasome, called M2, inflammasome have been associated. In fact, yes. So, and I mentioned about some drug that people, identify, to inhibit this pathway, the inflammasome. So there are drugs called dazufiram, and that is a drug that is used for prevention of alcoholism, the alcohol abuse. So that drug, it's a FDA approved, it's a very good. And then it was discovered that that drug also inhibit, gasdermin D. Actually, we published a paper last year in Science Immunology, which we use that drug, and we show that it inhibits the maturation, the cleavage of gasoline D and gasoline E. In fact, now that drug, it's being, to prevent the cytokine storm. It's being used in a clinical trial, I think in Brazil, in a clinical trial to, for COVID-19 to see if the cytokine storm that you mentioned can be, managed or, inhibited. Absolutely. So the pathway is very, very involved in, the cytokine storm. Then this will absolutely lead to organ, systemic effect that you mentioned. Yeah.
Thanks to everyone who can stay. If you need to leave, we understand. But Mary, I'd love to hear your question.
Oh, really beautiful talk, Gabriel. Impressive work. My question was, you showed nicely that gasdermin D is upregulated during osteoclastogenesis, but also that the pores don't assemble. So do you think that gasdermin D has an alternate function in osteoclasts?
Yeah, that is a good question. We really don't know. And yes, I mean, we have been really struggling. We have this, it didn't make sense. So, and we have some data now. Yes, it has nothing to do with just gasdermin D. It's downstream, like you were saying. Does it have something feedback with inflammation from? We don't know, but something we are suspecting based on some data that we are getting that maybe gasdermin D, maybe independently. I'm just saying, actually, inflammasome independent function of gasdermin has been shown. So is somehow gasdermin D involved in osteoclast death and independently of inflammasome? That is something that we are exploring now. We have some data, but it's just too preliminary to draw a conclusion.
That's really fascinating, though.
Yeah.
Thank you.
Thank you. Yeah.
I have a couple of quick questions. Really nice talk, Gabriel.
Thank you.
So one of your slides, you showed that two genes, S100A8 and A9, which comprise calprotectin, are somehow involved in the effects of the inflammasome. I wasn't really clear on what their role is thought to be.
Yes, I mean, absolutely. So S100, A8, 9, or yes. So these are, I said, this part of, the pyroptosis, I mean, when they are undergo pyroptosis. So the gasdermin, what they do is really more of, things like maybe prostaglandins may go through. But I think what is really known is about the cytokine R1 beta 18. So now those alarming that you say is more when cells are dying, because really what happens in a pyroptosis is cell rupture. The plasma membrane just rupture. And there is another protein that is involved downstream of the inflammasome. It's called NINJ1, which, and actually just rupture the membrane. So what I'm saying is now in that context, yes, people have shown that, many inflammatory factors, as you will expect, I mean, if they are produced at that time, then they will be just released to the environment, to the cellular milieu. And yes, yes, it will be secondary to, but not going through the pore body because they are dying. But again, the difference with apoptosis is believed to be silent. So say die, the plasma membrane is conserved, so things are not released, and then they are phagocytosed by what immune cell. But in the case of pyroptosis, it's because things are just released to the environment. It's just lies, and this is why it's inflammatory, because it propagates inflammation, as opposed, for example, to apoptosis.
Yeah. Okay, thank you.
And maybe I'll take the privilege of asking, we had another seminar speaker, which was Michelle McDonald, and she spoke about her work on osteomorphs. And I wonder if you envision that these pathways might be part of the process by which the osteoclasts change their identity and nucleate or divide.
Yes. So osteomorph, yes, a bit macrophage in the bone, and so yes, what do we think is, it's just, yes, we just try to understand the relationship between, osteoclast, this pathway with the osteoclast. And what we see is, again, really, the evidence is strong, but what we don't understand are the mechanism. So, I think this is one of the pathway, one of the mechanisms. So, I don't know for what reason. So, the osteoclast to do what you said, to fuse, to do whatever. So, but they don't want to be inflammatory. So then maybe they have to downregulate this pathway, which is again important for immune cell for the function to do whatever they have to do. I think for the osteoclast somehow this pathway and has to be done. And why? So I don't know. Maybe like we said. Yeah, they need to survive because this pathway are very inflammatory. They cause death, and maybe this is one way for the osteoclast to escape. It's to downregulate this pathway. Yes, so it's we don't know, but I think it's it's a possible.
Wonderful. Well, thank you so much, Gabriel. It was a great talk, and we were so glad to have you here with us today.
Thank you so much, Rudy, for the invitation, and I will stay in touch. Thank you very much.
Thank you.
Okay, thank you.